In this lecture, we are going to discuss the application of stable and radioactive isotopes in biological research and medicine. So, at the very beginning, let's define some terms. Isotopes are variants of a certain chemical element. Such variants, which have the same atomic number or proton number, but due to the difference in their neutron numbers, they are characterized by different mass numbers. The origin of the name isotope is related, it comes from this Greek expression, which refers to the fact that due to their identical proton numbers, they are displayed in the same position of the periodic table. We are going to talk about two different kinds of isotopes, stable and radioactive isotopes. And you have to keep it in mind that there are these two kinds of isotopes, which and many students have the wrong idea that whenever you hear the term isotope, it by definition means radioactive isotope, and that's a wrong impression. Um, and as you are going to see in a minute, not only radioactive but also stable isotopes have certain applications in biological research and medicine. So although in most of the lecture we are going to discuss the application of unstable or radioactive isotopes, in the first couple of minutes I'm going to summarize what you have to know about the application of stable non-radioactive isotopes. Since these isotopes are non-radioactive, therefore their presence cannot be detected by radiation detection because they don't emit any kind of radiation since they are stable isotopes. So if we want to detect the presence of a stable isotope which is different from the most frequently found isotope in nature, then the presence of the stable isotope can be detected by the mass difference. So here you can see a couple of such stable isotopes which are commonly used in biology and medicine. So for example, deuterium, which is the hydrogen isotope with a mass number of 2. So if you label a molecule with this variant of hydrogen, then and you compare it to the molecule which has only conventional hydrogen with the mass number of 1, then the molecule having one deuterium instead of conventional hydrogen will have one extra mass unit, and this extra mass, the mass difference, is detectable, and this forms the basis of detecting the presence of stable isotope in any kind of sample. There are several different approaches for detecting this mass difference. These are listed here at the bottom of the figure. Um, Isotope effect is the first principle based on which we can detect the presence of a stable isotope. The isotope effect refers to subtle alterations in the chemical properties of isotopes. And this is somewhat remarkable because in practically every case, we emphasize that the chemical properties of isotopes are identical because the chemical properties are determined by the number of electrons in the atom or in the isotope. And since they have the same number of electrons, therefore we expect their chemical properties to be perfectly identical. The fact that the isotope effect exists implies that although the chemical properties of isotopes of the same element are actually very similar to each other, there are subtle differences in their chemical properties, and this is what is referred to as the isotope effect. And this is where the most pronounced in the case of hydrogen 2, because the mass of hydrogen 2, deuterium, is 2 times larger than the mass of conventional hydrogen with a mass number of 1. So the fold difference between the isotopic variance of elements is the largest for the difference between hydrogen 1 and hydrogen 2. The other kind of approach with which we can detect the mass difference is density gradient centrifugation. And in addition, there are two different kinds of spectroscopic approaches: mass spectrometry, which detects the mass or the mass to charge ratio of isotopes, and spectroscopy, mainly infrared spectroscopy, which can be used for detecting the mass difference. Uh, but the principles of the spectroscopic techniques are not to be discussed in this lecture. The only example for the application of stable isotopes is the urea breath test. The aim of this method is to detect the presence of a bacterium, Helicobacter pylori, in in the stomach of patients suffering from gastric ulcer. Gastric ulcer is sometimes caused by this bacterium, and if it is indeed caused by this bacterium, then the ulcer is to be treated by antibiotics. Therefore, it is very important to diagnose whether a bacterial infection is indeed present in the stomach. And the stomach presents a very special environment for a bacterium, and it's very challenging for the bacterium to survive under these harsh conditions, because as you probably know, the pH of the stomach is quite acidic, and bacteria are typically killed by at this acidic pH. Helicobacter pylori is a notable exception to this rule, and it can survive under these harsh conditions, partially due to the presence of an enzyme, an enzymatic activity in the bacterium. The enzyme urease breaks down urea, and carbon dioxide and ammonia are produced as the breakdown products of urea, and ammonia uh, partially neutralizes the acidic pH around the bacterium, and this is how this enzymatic activity contributes to the survival of the bacterium. And this is the enzymatic activity with which the presence of the bacterium can also be detected. Because if the patient receives uh, urea in which is labeled by carbon 13, which is an isotope of non-radioactive stable isotope of carbon, and if this patient has Helicobacter pylori infection in his or her stomach, then the enzyme, which is only present in the stomach if the bacterium is present, then so the enzyme will break down urea, carbon dioxide and ammonia will be produced from this urea, and carbon dioxide enters blood vessels, and in this way, with the circulation, it will reach the lungs, and the patient will exhale carbon dioxide. And since the urea was labeled by carbon 13, therefore carbon dioxide will also contain this isotopic variant of carbon. So it is going to be carbon 13 carbon dioxide. And if carbon-13, carbon dioxide, is detected in the exhaled air of the patient, which can be achieved either by mass spectrometry or infrared spectroscopy, then it will be known that such an enzymatic activity is present in the stomach, and this directly implies that the bacterium is present, so the ulcer is caused by this bacterium, so it is to be treated by antibiotics. So the advantage, as you can see in the lower right corner of the figure of this method, is that it is non-radioactive. So the patient is not exposed to any kind of ionizing radiation. In the next and longer part of the lecture, we are going to discuss the application of unstable or radioactive isotopes. And as you can see in this introductory slide, I have divided the application of radioactive isotopes into four categories. First, we are going to briefly discuss the application of radioactive isotopes in vitro laboratory tests, which are aimed at the determination of very low concentration of analytes. And the name of this approach is radioimmunoassay. In the second and third uh, category, we are going to discuss the application of radioactive isotopes for studying biochemical processes by following the fate of certain radioactively labeled metabolites. And basically, the same principle is used in category three when the fate of such radioactive labeled substances called radiopharmaceuticals is followed in a whole animal or in patients in vivo diagnostic imaging. And at the very end of the lecture, I am going to talk about the application of radioactive isotopes for radiation therapy of cancers. First, let me introduce you to the principle of radioimmunoassay. And the aim of this approach is to detect the presence of certain substances or analytes in a certain fluid, which can be a serum sample. And let's assume that our aim is to measure the concentration of human growth hormone, these gray circles, in a human serum sample. So in order to detect human growth hormone in human serum samples by direct radioimmunoassay, we need at least two different kinds of antibodies against the same analyte. In my example, two different kinds of antibodies against growth hormone. One of the antibodies is called capture antibody. In a couple of minutes, you're going to see why it is called a capture antibody. And we need another kind of antibody, a detecting antibody. And these two antibodies must be able to bind to the analyte, growth hormone, at the same time. So they must recognize non-overlapping parts of the analyte so that they can bind simultaneously to the growth hormone. In this figure, red antibodies will be radioactively labeled, whereas this black antibody is not radioactively labeled. The first step in this procedure is the attachment of the first kind of antibody, the capture antibody, the non-radioactive capture antibodies, to the bottom of the plastic plate. And then the sample is poured into the plate. Uh, at the bottom of the plate, the capture antibodies are already present, and these capture antibodies capture or bind the analyte against which they were raised. And now you can actually see why this antibody is called a capture antibody, because it captures or binds the analyte. And the next step is to introduce something which is detectable, because only the radioactive antibodies are detectable due to the emission of the ionizing radiation. So we can add the second kind of antibody against growth hormone, the detecting antibody, to the plate. And the number of these red antibodies, so the number of the detecting antibodies binding to the plate, will be proportional to the number of growth hormones bound by the capture antibodies. Alternatively, or in actually more in most cases, the detection step is not done as shown here in the middle of the figure, but it is done in two consecutive steps. First, another anti-growth hormone antibody, if the analyte is growth hormone, is added, and this is not radioactive either. And then a secondary antibody is added, which will recognize this blue antibody, and this secondary antibody is radioactively labeled. But the principle is basically the same. The number of these radioactively labeled secondary antibodies will be proportional to how many growth hormones have been bound by the capture antibodies at the in the first step. So the principle is the same. The measured radioactivity, basically the number of bound radioactive antibodies, will be proportional to the amount, to the concentration of the analyte in the original sample. And let me come back to this part of the figure for a short time, because I would like to explain to you the principle of the secondary labeling, because that's the name of this procedure. So here we have this blue antibody is an anti-growth hormone antibody, which is able to bind to human growth hormone. And let's assume that this growth hormone, anti-growth hormone antibody was produced in a rabbit by immunizing a rabbit with human growth hormone. And then the red antibody here, which is called a secondary antibody, is an anti-rabbit antibody, which is able to recognize uh, antibodies produced in a rabbit, because another kind of animal, let's say a goat, has been immunized with rabbit immunoglobulins. And as you can see from this figure, the aim of secondary labeling is to increase the signal, because not only one, but typically two or three secondary antibodies can bind to a primary antibody, which augments or amplifies the signal. In this figure, then you were introduced to direct RIA, or direct radioimmunoassay. And in this figure, an alternative approach, indirect or competitive measurement, competitive radioimmunoassay, will be explained. The, the principle or the aim of this approach is the same. We would like to measure the concentration of something, let's say growth hormone, in the human serum sample. The first step in the procedure is identical to the first step in the previous approach. So we attach anti-growth hormone antibodies to the bottom of a plastic plate. But the second step is already different, because we pour a mixture into this plate, a mixture of the sample to be analyzed and the radioactively labeled version of the analyte. So if our aim is to measure the concentration of human growth hormone, then we need the radioactively labeled version of human growth hormone, which can be purchased somewhere. Uh, and then two different cases are shown in the middle of the figure. So what happens if the concentration of growth hormone is low in the serum sample we would like to analyze? The radioactive growth hormone and the non-radioactive growth hormone present in the original sample compete for the same limited number of binding sites. And if the amount of human growth hormone, the concentration of human growth hormone in the serum sample is low, then a relatively large number of radioactively labeled human growth hormone will bind. If, on the other hand, the concentration of human growth hormone is large in the serum, then this large concentration of non-radioactive growth hormone will compete with the radioactively labeled version for the same binding sites. And since the non-radioactive growth hormones outnumber the radioactively labeled growth hormone molecules, therefore, relatively few radioactively labeled growth hormone molecules will bind to the capture antibodies. So basically, there is an inverse relationship between the amount of bound radioactivity bound to the capture antibodies, and the concentration of analyte in the original sample. So if we finally measure the radioactivity, there will be an inverse relationship between the concentration of the analyte to be determined and the measured radioactivity. In this part of the lecture, we are going to talk about how we can study biochemical processes and physiological or pathophysiological processes in cells using radioactively labeled metabolites or radioactively labeled molecules. The principle of these measurements or of this approach is that if we label a molecule, a metabolite, glucose, or any kind of amino acid with a radioactive isotope, then this radioactively labeled version of the molecule behaves in an identical way to how the non-radioactive counterpart behaves. So if we follow the fate of the radioactively labeled version, then we can basically learn how the non-radioactively non-radioactively labeled version of the molecule behaves in a living cell or in an animal. And since the radioactive isotope will emit ionizing radiation, the presence, the location of the radioactively labeled metabolite can be detected or can be traced in the human body or in the cell culture or tissue culture. The radioactively labeled biological or pharmacologically important molecules are named radiopharmaceuticals if they are applied in medicine, and if they are applied in biological research, then they are usually called radioactive tracers. The whole approach, which may sound trivial nowadays in the 21st century, but was actually a breakthrough in the middle of the 20th century. And this approach was pioneered was a Hungarian scientist called George Hevesi, who received the Nobel Prize for his achievements in this research in 1943. And at the bottom of the figure, you can see a couple of examples for what kind of radioactive isotopes can be used as tracers. Um, you don't have to know these examples. These are just for demonstrating the principle. As you can see, basically any kind of a metabolic process, let it be amino acid metabolism or DNA synthesis or the functioning of ion pumps. So basically anything you can think of can be followed or traced by the application of radioactive isotopes. As I said, uh, radioactivity is required for making the whole process visible or detectable, and the approach with which the presence of the radioactivity is measured is called autoradiography. And this technique the uh, involves the detection of radioactivity by a film or nowadays a detector, which is placed on the sample to be investigated. And this sample to be investigated can either be a cell culture sample or a tissue section, or if some kind of a blotting or electrophoresis followed by blotting has been carried out, then it can be the blotting membrane itself, and the presence of the radioactivity will be actually shown by the film or the detector. In the next part of the lecture, we are going to talk about the application of radioactive isotopes for medical imaging. And in the first slide, I would like to talk about what kind of considerations you have to make when you want to select a certain isotope for medical imaging. One of the things you have to consider is the type of the radiation emitted by the isotope. And as a general rule, only isotopes leading to directly or indirectly leading to the emission of gamma photons or X-ray photons. So only these kinds of isotopes can be used for medical imaging purposes. So these are the 5 different kinds of radioactive decays we have previously talked about. And as you can see, neither alpha nor negative beta decaying isotopes are optimal for medical imaging because the path length of alpha and negative beta particles is too short in the micrometer or millimeter range. Therefore, if such an isotope is present in somewhere inside the body, the particle, the alpha or negative beta particle emitted by the isotope will not get out of the body. Therefore, it cannot be detected by an external detector. On the other hand, these 3 different kinds of radioactive decays can be used for medical imaging. Positive beta decaying isotopes lead to the emission of a positron, and this positron undergoes annihilation, which leads to the emission of 2 gamma photons. And these gamma photons, so not the positron itself, which was originally emitted by the isotope, but these 2 gamma photons produced by annihilation are actually detected in a technique called positron emission tomography, abbreviated PET. Electron capturing isotopes or K capturing isotopes can also be used for imaging because after K capture, a vacancy is created on the K shell, and this vacancy is filled from a higher shell, which is followed by the emission of characteristic X-ray, and it is this characteristic X-ray photon which is detectable. And obviously, gamma decaying isotopes, since they produce gamma photons, can be used for medical imaging. The second consideration you have to make when selecting an isotope for imaging is the physical half-life. The physical half-life is of an optimal isotope as far as medical imaging is concerned, is a couple of hours. And so it must not be too long or too short, because if the half-life of the isotope is too short, let's say a couple of minutes, then it is impossible to complete the whole measurement before all the radioactivity decays. Because if the half-life of an isotope is, let's say, 1 minute, then in 10 minutes or 20 minutes, there is practically no radioactivity present because all of the radioactive isotopes have already decayed. And this doesn't give us enough time to complete the whole procedure because that's a relatively long procedure. The radiopharmaceutical has to be synthesized, it has to be injected into the patient, and the measurement has to be carried out. And this is a relatively long process. If the isotope is too stable or so decays very slowly, then the half-life is very long. And for reasons not discussed in detail here, too large amount of radioactivity must be injected into the patient so that a reasonable number of decays take place during the imaging process, and that's basically dangerous and therefore cannot be, it is not acceptable. And the final thing you have to consider when selecting isotopes for imaging is the biological half-life, because the biological half-life must match the time scale of the measurement uh, for reasons very similar to the considerations we made with the regarding the physical half-life. So if you are looking at a process which takes place on the hour scale, and the biological half-life of the isotope is a couple of minutes, then you simply cannot look at the process you're interested in, because by the time the process happens, or during the most interesting part, when the process happens, the radioactive isotope has already left the body by biological decay. This table, as you see in the upper right corner, is supplementary material. So the actual examples are not to be known, but you can see that different kinds of isotope can be used for different kinds of purposes. And the other relatively important message of this figure is the importance of this isotope, radioactive metastable technetium-99, which is the most widely used isotopes because, as you can see here, it can be incorporated into a wide range of radiopharmaceuticals, which make it a very versatile radioactive isotope for medical imaging purposes. In this figure, I would like to explain to you uh, what the optimal properties of radioactive metastable technetium are, and how it can be used for how it can be produced and used for medical imaging. As you can see, radioactive technetium, metastable technetium, uh, decays by gamma decay with an optimal half-life of 6 hours, and a stable technetium is produced. So the term, the letter M here means metastable, and the term metastable means that it's an unstable isotope, that it will undergo gamma decay with an optimal half-life of 6 hours. That's a very convenient half-life, not too long, not too short. It gives us enough time to synthesize the radiopharmaceutical and to carry out the whole procedure. Another optimal property of radioactive metastable technetium is its mother nucleus and the process in which the mother nucleus leads to the production of radioactive technetium itself. Because as you can see, the mother nucleus produces metastable technetium with a radioactive, with a negative beta decay, with a relatively long half-life, 66 hours. And this means that the mother nucleus can be stored for a relatively long time. And in this way, uh, radioactive technetium is produced from the mother nucleus for a relatively long time uh, in a device called technetium generator, which is going to be shown in the bottom of the figure. So the salt of the mother isotope, uh, molybdate, which has a double negative charge, is bound to alumina, aluminium oxide, and due to the presence of this double negative charge, the binding of the salt to the column is relatively strong. When the mother nucleus undergoes a negative beta decay, technetium is produced, and the salt will be present. So pertechnate is so molybdate is converted to pertechnate, but pertechnate is only has only one negative charge, and therefore it binds less strongly to the column, to alumina, and therefore it can be relatively easily eluted by pouring saline through the column, and in this way radioactive technetium in the form of a salt is produced, and from this salt the isotope can be incorporated into radiopharmaceuticals, which can be used for any of the procedures which were highlighted in one of the previous figures. In this figure, I'm going to try to explain to you the principle of the gamma camera, which is the simplest device used for detecting the presence of a radioactive isotope in the human body. You can see a diagram or a schematic representation of the device in the upper left part of the figure. Let's assume that a radiopharmaceutical accumulating in the heart has been introduced or injected into the patient, and therefore the gamma photons are mainly emitted from the heart. The first component of the device is called the collimator. A collimator is a sufficiently thick absorbing lead slab into which a large number of holes have been drilled. These are the holes, And as you can see, the gamma photons can only go across the collimator if they are parallel to the holes. If they are not parallel to the holes, then they collide into the wall of the holes. And since the collimator is made of lead, which has a relatively high absorption coefficient for gamma radiation, these gamma photons will be absorbed. So the result is that only this part of the detector of the camera will receive gamma photons, which the part which is above the accumulation place of the gamma photons. And this second part of the device is the scintillation crystal, in which the gamma photons produce scintillations or light flashes, so visible photons, and these visible photons are detected by photomultiplier tubes. And this, the right-hand side of the figure demonstrates the importance of the collimator, because here from this device the collimator was removed. And this shows what would happen if there were no collimator in the device. Gamma photons are emitted in all different directions. And in the absence of the collimator, all of these gamma photons are able to reach the scintillation crystal. Scintillations or light flashes are produced all over the scintillation crystal, and therefore all of the detectors will detect the light flashes, and all of them would imply that that they detect radioactive or ionizing radiation. So in the end, without the collimator, we wouldn't be able to locate the signal. So this shows that the collimator is actually an essential part of the device. But a large disadvantage of the collimator is that most of the gamma photons are actually absorbed because they are parallel to a hole only very rarely. Therefore, only a tiny fraction of the emitted radiation is actually detected. But we have to be able to live with this disadvantage, because that's the only way how we can locate the signal, as shown by if you compare the real gamma camera with the one from which the collimator has been removed. Let me show you a couple of examples for what kind of images you can produce with the gamma camera. In the left part of the figure, you can see the application of radioactive metastable technetium for imaging the kidney. And we not only produced an image, a static image of the kidney or of the kidneys, but the kinetics of the accumulation of the isotope, so the accumulation of the isotope as a function of time, has also been measured. And on the right side, you can see bone scintigraphy with another kind of technetium-based radiopharmaceutical. And the aim here was to detect bone metastasis from prostate cancer. If cancer metastasizes into bone, then at the site of the metastasis, very active bone restructuring or rebuilding is taking place, and therefore this radioactive pyrophosphate, technetium-labeled pyrophosphate, will be incorporated into bone, and if high amount of incorporated pyrophosphate, pyrophosphate is present, that then it implies that very active bone rebuilding is taking place, and the most likely explanation for this is a cancer metastasis. If you look at this and these two images, you may have the impression that the resolution, so the detailedness of these images is not perfect, to say the least. I guess many of you have already seen conventional chest radiographs, for example, X-rays, chest X-rays, and the resolution of such a conventional chest X-ray is superior to this image, to the gamma camera image produced on the right. So you may wonder why we actually do this kind of gamma camera measurement if the quality of the image produced seems to be inferior to what can be produced by conventional X-ray. So it's a fact that the spatial resolution of the gamma camera images is actually poor. But actually, but when producing gamma camera images, we are not really interested in the morphology or in the anatomical details of the organs, but we would like to measure the metabolism, the blood flow, different kinds of biochemical processes, and this can be quite accurately performed with gamma camera imaging. And this is called functional imaging. So gamma camera is a functional imaging modality because with its application, our aim is to image the function of an organ or of a tissue and not the anatomical details of the tissue or the organ. As you may have seen in the previous slide, images produced by a gamma camera are two-dimensional images. So the third dimension, the depth information is lost. So we cannot say uh, at which depth of the human body the radioactive isotope accumulates. We can only see a two-dimensional projection image. Obviously, this is a disadvantage, and the three-dimensional version of the gamma camera has been developed, and this tomographic or three-dimensional version of the gamma camera is called SPECT, standing for Single Photon Emission Computed Tomography. And the principle of SPECT is very similar to the principle of X-ray absorption computed tomography, so the mother of all tomographic techniques. And therefore, I'm not going to discuss the details of the imaging principles, but very briefly, uh, different gamma camera images are recorded at different angles, and this is achieved by either having one single gamma camera and this single gamma camera producing several or recording several images from different directions of the same patient, or in more developed SPECT devices, there are more than one gamma cameras, two or three gamma cameras, and they capture an image, or if there are two gamma cameras, they capture two images at the same time. Then both gamma cameras rotate around the body and they capture another pair of images at another angle, and this is repeated a large number of times. And using the same kind of image reconstruction principle, which is used for producing X-ray computed tomography images. So basically, that's the principle of back projection. the three-dimensional distribution of the gamma-emitting isotope can be reconstructed. There is another approach called positron emission tomography, in which uh, detectors on opposite sides of the human body are present. Uh, and positron emission tomography is based on the detection of the two gamma photons produced by annihilation after positive beta decay. And the detection in the positron emission tomograph is based on the coincidence detection of these two gamma photons colliding to detectors on the opposite side of the human body. Such a coincidence detection is not present in SPECT, not even when two or gamma cameras are present in on opposite sides of the human body. So coincidence detection only applies to positron emission tomography. In this image, I would like to show you a couple of example image series for what can be achieved with SPECT images. In the upper part of the figure, you can see horizontal sections of the human brain, which has been labeled by this radiopharmaceutical. You don't have to know the name because that's not important for you right now. This radiopharmaceutical is used for imaging brain perfusion, so how much blood basically flows across a certain part of the brain. And the warmer color and the corresponding to the cortex, to the cortical region, is typically characterized by higher blood perfusion, and that's the normal case. So that's in the upper part of the image. You can see the blood perfusion typical for a healthy human brain. And in the bottom image series, you can see the same kind of perfusion images, but in this case, the images are from a patient suffering from Alzheimer's disease. And these images are characterized by cortical hypoperfusion, and this kind of feature is characteristic for patients from suffering from Alzheimer's disease. So this demonstrates that such a kind of functional modality or functional information regarding brain activity or brain blood perfusion can be produced by SPECT. And I would like to again emphasize that we are not really interested in the anatomical detail or the morphology of the brain when performing SPECT. We are rather interested in the function, in this special case, the blood perfusion of different parts of the brain. And finally, I would like to explain to you how radioactive isotopes can be used for radiation therapy. The purpose of radiation therapy is to treat tumors. We can classify the application of radioactive isotopes for radiation therapy based on the location of the radiation source, which can be inside or outside the body. First, let's discuss what kind of radioactive isotopes can be used when placed in the human body, typically somehow close to the tumor to be treated. The first example is for this class is alpha emitting isotopes, and the path, the penetration depth of an alpha particle is typically on the micrometer scale, and therefore In order for such an alpha decaying isotope to be therapeutically useful for treating tumors, this alpha decaying isotope must be must be present very close to the tumor on the cellular scale, basically, because as you can see, the path length of alpha particles is not larger than the size of a eukaryotic or human cell. Therefore, the alpha emitting isotope must be targeted very specifically. And here you can see one way of targeting an alpha decaying isotope if it is coupled to an antibody. And if such an antibody is injected into the patient, the antibody will bind to the primary tumor and also to the metastasis. And due to the presence of the alpha decaying isotope, the tumor is destroyed. In the bottom part of the figure, you can see the relative depth dose of an alpha particle, and you can see that this is characterized by a non-homogeneous uh, production of ions or ionizations, because for a relatively long part of the path, few ionizations are produced, and then suddenly the number of ionizations, so the amount of damage, biological damage delivered by the alpha particle, actually increases, and then the particle suddenly stops. So the most of the damage is actually done by the alpha particle very close to its path and very close to the end of its path. And this peak is called the Bragg peak, which is characteristic for alpha particles. And as you're going to see in the next slide, it also characterizes proton radiation. Another kind of isotope, which is typically used for radiation therapy when the radiation source is inside the body, is negative beta radiation. But the penetration depth of negative beta particles is a couple of millimeters, and therefore topical application is required. So the isotope must also be present to relatively close to the tumor. Um, this is in many cases achieved by brachytherapy, in which the isotope is placed onto or into the tumor in a capsule, and this is achievable in the case of tumors which are surgically relatively easily accessible, so they are relatively close to the surface, to the skin. So this principle applies, for example, to cervical or prostate cancer, skin, and breast tumors. And the red curve at the bottom of the figure demonstrates the, the ionization density produced by the beta particle. And you can see that this is quite evenly distributed all throughout the path of the beta particle. The second class or the second type of the application of different ionizing radiations for radiation therapy is when the radiation source is outside the human body. Such a radiation can be, for example, electron radiation. This electron radiation the absorption and the ionization properties of this electron radiation are practically identical to the properties of negative beta radiation, because negative beta radiation practically consists of electrons. But there are notable differences as well due to the fact that this electron radiation is produced by particle accelerators, and by adjusting the voltage of the particle accelerator, the electron beam, the energy of the electron beam is also adjustable. And as you can see at the bottom of the figure, the penetration depth of the electron radiation is determined by the energy of the electrons. So if 3 million electron volt, the kinetic energy of the electron is 3 million electron volt, then the penetration depth is approximately 1 centimeter. If, on the other hand, the kinetic energy of electrons in the electron radiation is 20 million electron volts, then the penetration depth is approximately 5 to 6 centimeters. In this way, we can adjust how deeply the electron beam penetrates into the body, and therefore it can be controlled. Therefore, the energy of the electron beam can be adjusted to how deep the tumor to be treated is under the skin. You can see, however, that the damage which is done by the electron radiation is continuous. So if the tumor is, let's say, 5 centimeters under the skin, and this kind of an electron beam is used for its treatment, then the healthy tissue, which is 2 centimeters under the skin, will also be damaged by this electron beam, because as you can see from the more or less horizontal line, the same amount of ionization, so damage, is actually delivered to the healthy tissue as well. There is a way to get around this problem, which can be achieved with the application of proton radiation. The principle of how proton radiation is absorbed, how it produces ionizations, is very similar to what we discussed for alpha radiation. So the ionizations produced by proton radiation are also characterized by the presence of the Bragg peak, as explained for alpha particles. But due to the fact that this proton radiation is produced by accelerators, these protons have adjustable energies. And since the position of the Bragg peak is determined by the kinetic energy of the proton, we can fine-tune how deeply the radiation penetrates, so we can adjust the energy of the proton beam to the location of the tumor, which is nicely demonstrated in this figure at the bottom, by this graph at the bottom of the bottom of the figure. So if the tumor to be treated is 5 centimeters under the skin, then a 100 million electron volt proton beam can be used. If, on the other hand, the tumor is 12 centimeters under the skin, then a proton beam with a kinetic energy of 150 million electron volts is to be used. You can also see that if the tumor is 12 centimeters under the skin, and we use this proton beam shown by the black line, then the dose delivered to the healthy tissues closer to the skin is much less than the dose which is delivered to the tumor, and this is due to the presence and the characteristic presence of the Bragg peak, which is typical for protons and proton radiation and alpha particles. A significant drawback of proton therapy is its price. So such proton accelerators are extremely expensive, and this has to do with the fact with the very large kinetic energy of the proton. Therefore, very few such accelerators exist in the world currently. The most common type of external radiation source produces gamma radiation or X-ray, and in most cases, actually, gamma rays are used in most cases for radiation therapy. The name of this therapeutical approach is cobalt therapy, which is a misnomer, as you can see in the scheme in the middle of the figure, because cobalt is only the mother nucleus, which decays to nickel, and the gamma photons used for treating the tumor are actually produced by the metastable nickel isotopes. You must know about gamma radiation that it is absorbed according to an exponential absorption law, which has deleterious consequences for how it can be used for treating tumors. Because just imagine if a tumor is 4 centimeters under the skin, then these tissues, 1 to 2 centimeters under the skin, are healthy tissues, but you can actually see that they get a higher dose than the tumor itself. So the conclusion from a medical perspective is that selective irradiation of tumors is impossible if we only apply a single gamma radiation source due to the exponential absorption law of gamma photons. In order, in order to get around the problem of the lack of specificity of cobalt therapy, uh, different kinds of approaches have been introduced, and the basic idea of getting around the problem introduced in the previous figure is to apply many different beams, all of them aimed at the target, at the tumor. One realization of such an approach is called the gamma knife. In this application, approximately 200 gamma sources are incorporated into this helmet. Um, this is typically used for brain surgery, and all of these gamma sources are arranged such that all of the beams are aimed at the tumor. Individually, these beams are relatively weak, but since all of them are aimed at the tumor will receive will be hit by all of the beams, and therefore the damage delivered to the tumor is relatively large. Therefore, the tumor will be destroyed. On the other hand, since the individual beams are relatively weak, the healthy tissue surrounding the tumor will not damage to a large extent because the healthy tissue is only affected by a single relatively weak beam. Therefore, the healthy tissue is preserved and only the tumor is targeted specifically. And actually, the word knife in the name of this technique implies that it is almost as accurate as the knife or the scalpel of the brain surgeon.